This is the fourth lecture for Neuroscience A. Today I'm going to talk to you about signal transmission, the first part of the lecture about how neurons communicate with each other in the brain. And I'll put this video up in a couple minutes when I'm done. Today's goal is to start to understand how information is communicated between neural cells in the brain or other parts of the nervous system. Specifically, we're going to learn how signals are conducted electrically. And so today there's two main concepts that we're going to cover. One is what's called resting membrane potential. So it's the membrane potential is the main concept. It's the potential or voltage, right? Voltage difference of the inside and the outside of the neuron cell. So resting is basically when the cell is not doing anything. What is the voltage of the cell? And then second, we'll cover what's called action potential, which is the main way that the neurons use to signal each other in the mammalian brain and bird brain and pretty much all large animals other than certain special types of neurons in humans. So today I'm going to cover basically the electrical signal transmission. How does action potential and membrane potential change in neurons, and how is it transmitted along the axon? And then next week, I'm going to cover chemical signal transmission. So actually, when the signal travels along the axon, when it reaches the end of the axon, and it needs to be communicated to the next neuron, the postsynaptic cell, it actually uses chemicals, neurotransmitters or hormones, things like this, to communicate the signal. So that's going to be covered next week. This week I'm just covering the electrical signal transmission, which is going to be the action potential. And to understand action potential, we have to understand what is membrane potential and what is resting potential. So today the two main topics are the resting membrane potential. And within that, I'm going to cover a couple of different subjects. I'm going to introduce you details about cell membrane, and I'm going to remind you about diffusion and the electrical force and how those result in there being a potential difference across the membrane. Then I'll introduce the equilibrium potential for different types of ions. And we'll cover the different concentrations that actually are inside and outside most neural cells and that cause the actual membrane potential of neural cells to be what it is. Second, I'm going to cover action potentials, which is basically a very fast change in membrane potential. And the way that action potentials work is basically you have what are called voltage-gated channels. Basically special channels that open or close when the voltage changes. So you get a kind of feedback loop where the membrane potential keeps rising or falling, and it feeds itself to keep going faster and faster and faster. And you get this shape that's, if you have time going like this and the membrane potential V going like this, you get this shape that looks like this, where it changes very quickly. It goes up and then back down. And that's why it's called a spike or an action potential. It looks kind of like a spike in the membrane potential. So that's the second thing I'll talk about is voltage-gated channels. Finally, at the very end, I'll cover a couple of just one slide each. I'll cover toxins that affect the nervous system via channel blockage. I'll talk about conduction with myelin, why we have myelin and how it changes the conduction. And then I'll finally tell you about a different way of inciting membrane changes, which is optogenetic. It's a more recent development since the year 2000, the last 20 years, basically. Okay, so what do you know about how signals are transmitted? Well, you might think that maybe the most common is a sort of copper wire or phone wire, or maybe fiber optic wire, right? So I'm going to cover electrons and copper wire here. So this is a phone line, big, thick phone lines, right? And I'm going to contrast how the brain works with how copper wires, electrical signals, are sent in engineered human solutions. We'll look at this first. So in copper wire, the medium, in other words, how is the signal transmitted? Basically, electrons move, or, well, they push electrical signal along the copper wire, either continuously in direct current or back and forth in alternating current, right? But the speed is very fast, basically about half of the speed of light, which is about 150,000 kilometers per second, or 150 million meters per second. So very fast. And it's pretty well insulated. We put, it looks black because sometimes we'll have rubber around the wire. But even if we don't, actually the center of the wire, the outside of the wire will actually kind of insulate the center of the wire, in which the transmission will be the most, will be happening the most. So that's a phone wire. That's how engineered copper wire conducts a signal. Compared to that, neurons, the brain, are pretty inefficient. Basically, the medium of signal transmission is you have, some cell, and inside of it you have a bunch of ions, molecules, atoms, right? Ions. And outside of it you have some ions. And the signal is transmitted by the ions moving into and out of the cell, and that makes the signal transmit along the wire, or sort of a fiber, right? It's relatively slow, only about 10 meters per second. So you see that that's very slow compared to this 1.5 millionth of the speed. Sorry, 1/15 millionth of the speed. And it's pretty leaky. Obviously, if you have ions in a sort of soup, it's they're going to go wherever they want to go. They're not going to be bound to the copper wire like electrons are, because there's air around them. It's air, so it's not going to transmit current very well. So the first topic I'm going to introduce is membrane potential. And the easiest way to understand it is what is resting membrane potential. Resting when the cell is not doing anything. So first we need to understand what is inside and outside of a neural cell, right? So I already drew a picture. I drew a picture that's kind of, we have a, I'm going to draw the neuron cell. Kind of looks like this, right? Here's the axon going away. These are the dendrites. This is the cell body, with the nucleus and stuff. And this is the axon. And basically the cell it's got like a skin. I drew it as this red thing. It's 3 dimensions, right? I'm just drawing 2. This red thing is kind of like a skin, which is called, I don't want to do that. It's a membrane, right? Is the skin of the cell is the membrane. And inside you have basically water, right? Water is in here, and actually water is out here. You have some fibers, to control it, but it's mostly water. And in the water there's some ions, right? It's not just pure water. It's mostly water, but inside the water there's dissolved certain salts like sodium chloride, salt, table salt. Those are dissolved in the water due to good. Basically, water has special properties that makes it good at dissolving things, and so when you eat salt, you it's broken down and dissolved into the water into chloride ions and sodium ions. This is a crystal of salt, and then the water sort of breaks it down. And you can see that since water is polarized, it has sort of like a positive side to it and a negative side to it, just because of the way that a property of water that arises from the way it's formed out of oxygen and hydrogen. Because of that, you get these chloride ions and sodium ions dissolved in the water, and these are now ions, right? You have Na plus, so it has it is missing an electron basically, so it's positively charged, and you have a chlorine ion which is negatively charged. It basically has an extra electron. So that's from salt, table salt. Actually, you have other things in the water, dissolved in the water. Potassium ions. So from eating, bananas and things like that, it's very important. You'll learn for your brain's function. And also, there's calcium, Ca2+. So, these are both positively charged. Calcium has two fewer electrons, so it's double the positive charge, two positive charge units more. And these are also dissolved in the water, inside and outside of your brain cells. And I mentioned that so the cell is basically they have a skin, a membrane that holds everything inside, and it tries to keep everything outside, right? The same way that humans have skin, cells have skin. And the skin in cells is made from a bilayer of phospholipids, right? So basically fats, a special type of fats that are sort of waterproof. They push water away, it's hydrophobic. So basically it makes it's kind of a waterproof skin that keeps water inside and keeps the water outside. But I mentioned that there's ions inside and outside the cell, right? So how do they move between it? You can't magically teleport inside the cell. Well, the same way that human skin has pores, you have holes in your skin that let sweat and things go out. The same way that human skin has holes, pores, you also have in cells, you have membrane proteins embedded in the skin. And some of these basically are tunnels or holes in the skin, in the membrane, called ion channels or channels, through which ions can flow into or out of the cell. And that's how ions can move from outside to the inside or the inside to the outside. And I mean, just like everything in the body, these are basically just made out of proteins, specially formed, shaped proteins, made from basic amino acids that are formed into an alpha helix structure, and they're folded in some special way. And then these things that are called subunits are combined. Like there's 5 of them to make like a circle, basically a cylinder that has a tunnel in the middle that ions can flow through. So it basically makes a tunnel through the membrane. This is the inside of the cell, and this is the outside of the cell, right? The cell's like this, This is just part of its skin, part of its membrane that I'm visualizing here. So because of certain physical forces, chemical physical forces, the diffusion of ions and the electrical force, because of these forces, the neuron, the inside and the outside of the neuron cell will actually hold a potential difference, a difference in voltage, right? So if we measure the voltage difference, so basically we stick a wire called an electrode or a microelectrode. Micro, it's very small, right? Because the cell is, maybe 30 micrometers, across. Micrometers, 10 to the negative 6 meters, right? Very small. Oops. So we stick an electrode, a wire inside the cell, and we stick another wire, the ground wire, outside the cell, and we measure the difference in potential. Or actually, we measure the difference between these two wires. And usually, in most sort of brain cells you'll find in mammals or birds or things like this, you'll find that there's a negative resting potential. In other words, the inside of the cell, the inside is negatively charged. There's more negative ions inside there, or fewer positive ions, right, than outside the cell. And so this is called resting potential. Well, membrane potential, because it's the potential voltage that is felt across the membrane. So here's a zoomed in picture, right? We can see the potential across the membrane. And it's called resting because the cell is not doing anything. Basically, we just measure it when the cell is sort of not doing anything in particular. And this is caused by two specific physical forces that I mentioned. One is diffusion of ions through the channels, and the other one is just the electrical force that pushes ions through channels because of electrical fields. So first I will talk about ion diffusion. So first of all, if we didn't have any channels in the membrane, so no holes, no tunnels in the skin, then the situation would look like this, right? One side, wherever all the ions started, would have all the ions. The other side would just have pure water, whatever is in there, because nothing can travel between them, right? Nothing can travel because there's no tunnels, no pores or channels to travel through. But actually the situation, if you have some channels, some tunnels, these are called leakage channels because they let ions travel both directions. Leakage channels allow for transmembrane flow across. Trans means across or between, across the membrane. And if you will just have leakage channels, then basically ions will flow down the concentration gradient, right? So a basic concept in chemistry, physics, is that entropy always increases, right? You always have things flowing from areas of high concentration to areas of low concentration to make the concentration the same everywhere, right? And so if you just give some holes and you leave it alone, eventually you'll have the same number on both sides, right? The same number of sodium, the red ones, and the same number of chloride, the blue ones, right? The concentrations will be the same. because they'll diffuse down the concentration gradient. So this is what causes the resting potential because of ion diffusion. But you can see this wouldn't really hold a charge, right? This is the same amount on both sides. So it would be the same voltage on both sides. So nothing interesting would happen. Well, there's one other force that is also moving ions, and that is the electrical force. Basically, if we have the perfect membrane with no holes, no tunnels, no channels, then even if we put a battery, if we force a potential across the membrane, there will be no current. None of the ions will move because none of them can move, right? But if we give them the ability to move by giving them channels, then the electrical field caused by our potential difference will cause ions to move. Specifically, if we have the negative side over here, you have a negative charge over here, then negative charges will be repelled by negative charges. It'll push negatives away, right? And pull positives. Negatives are pushed away. And it will also pull positives, towards it. This is basic electromagnetic field. Same thing will happen for the positive side, right? It will push away like charged charges, and it will pull or attract different charges. So I don't like introducing new terminology. I know this is English's second language for a lot of you, but in English they say traditionally cathode. I mean, a lot of this is from German or Latin, but you say cathode is the negatively charged side of the battery, and the anode is the positively charged side of the battery. And we call negatively charged ions anions, right? Our negative ions. So our Cl minus is an anion. Cations are K plus or Na plus, sodium or potassium, and those are called cations. I'll just say usually positively charged ions or negatively charged ions, so you don't have to remember. Okay, so now I'm going to talk about, in this real specific case of neurons in the brain, what causes the resting membrane potential? Well, one aspect of it is caused by the equilibrium potential, the balance of potassium ions, positively charged potassium ions. And basically, for a reason I will tell you later, inside the cell, so inside is on the left here, inside the cell, you have amino acids and proteins, just stuff that is inside the cell to make the cell work, right? Signaling proteins and things like that. These are usually negatively charged. There's a higher concentration of those inside the cell. There's also a higher concentration because of a special ion pump. There's a higher concentration of potassium ions inside the cell. And because these negatively charged ions, the A minuses, things like big proteins. They're usually big, or because of their shape, they can't pass through special normal channels that the potassium can pass through. And so basically, these guys, they try to go through, but they can't. They're too big. So they can't go through. But the potassium do go through. They want to go down the concentration gradient, right? And so you end up with basically an equilibrium where when the potassium ions go out, right, they'll try to balance their diffusion, the concentration. But remember, the minuses pull the pluses. So actually, they'll try to go back in because this is going to be more negative because these guys are stuck inside here. So the balance, the equilibrium, the balance between the potassium trying to go out to equalize its concentration and the potassium getting pulled back inside because it wants to be attracted to the higher, so the negative voltage in here. That will balance. And the level that balance is at depends on how many of these there are, right? The relative concentrations. But usually in neurons in the brain, it's about negative 80 millivolts. So after both of these processes happen, you leave them for a long time, right? You let them achieve the equilibrium. It happens at about negative 80 millivolts. So the inside of the cell will have negative 80 millivolts, right? Millivolt is, remember, 10 to the negative third of a volt. So 1/1000th of a volt, right? And so because of this type of process, you end up with a voltage, a potential difference across the membrane. Because you have, so the negative guys stuck out here, attracting the positive guys stuck out here. So if you measure right, near there, or basically almost anywhere inside of here, you'll see a potential difference in voltage between the inside, cytosol, means inside of the cell, stuff inside the cell, and the extracellular fluid, which is the fluid outside of the cell. Of course, within this area, inside the cell, or outside the cell, it's usually electrically neutral. So there's equal amounts of a positive and negative charge. Because of course that's what physics wants to happen. But and then of course near the membrane you're going to have a sort of a concentration of positive and negative charge inside and outside. But if you integrate over the small space across the membrane, you'll have a equal number of positive and negative charge. So it all balances out. So first I talked about the potassium balance, K plus balance, right? But there's actually another balance. There's other ions in the cells and outside. And the next one I'm going to talk about is the sodium ion balance. And the situation here is the same as the potassium case, but it's kind of opposite. So basically, because of the ion pump that I'll explain in a moment, there's more sodium outside the cell. Lots of sodium outside the cell. And this concentration, right, if you allow it to go to travel through the cell, it will try to diffuse, to equalize the amount of sodium inside and outside. And there's other things outside here like chloride. There's like, chloride and stuff like that out here. And these are basically small special channels that only let sodium go through, or just general leakage channels also. And so sodium tries to go inside the cell to diffuse in there, but other ions in there like potassium and stuff like that, sort of lined up along the inside of the membrane, they balance out the diffusive force by keeping the sodiums outside the cell. And so the equilibrium potential for sodium is actually positive 62 millivolts. Right? So actually, when sodium is balanced inside and outside, when the inside of the cell has positive 62 millivolts, right? Potassium was balanced when it was negative 80 millivolts, right? Sodium actually has a positive equilibrium potential. You need to have sodium inside the cell when it gets to 62 millivolts, then it will be balanced, diffusion and electrical force. So what that means is if you leave the cell alone, it tries to raise the membrane potential up to 62. If you only had sodium and these other anions, right? If you only had sodium, the inside would be plus 62. If you only had potassium, the inside would be negative 80 millivolts. So that's what this equilibrium potential means. It's the potential that it tries to achieve. So you can see that these sodium and potassium are working in opposite directions, right? Sodium's trying to pull the membrane potential up. Potassium is trying to pull it down. And so these balance out also, right? Sodium, if more sodium goes inside, it will go up, which makes the potassium ones try to leave the inside of the cell faster, right? And so you'll get a balance, an equilibrium of all the different ones at the same time. That's what causes the resting membrane potential. It's the combination of all the different equilibrium forces and electrical forces together. And the resting membrane potential and, of course, the equilibrium potentials will change based on the concentrations inside and outside the cell. So you can see I've listed here for potassium, sodium, and calcium and chloride the different potentials. So you can see chloride causes it to go, it wants to go negative, so does potassium. Whereas calcium and sodium are causing it to go up. So basically these two are kind of cause excitation. They cause it to go up, right? Excitation. Whereas potassium and chloride, potassium leaves the cell, chloride enters the cell. They inhibit the activity. They make the membrane potential go down. So when you add all of these together, taking into account their relative concentrations, so calcium has a very low concentration, even though it has a higher, stronger pull, right? It has low concentration. When you balance all of these, you get about negative 65 millivolts. And the main thing that moves this is sodium and potassium. Those are the main things, right? So I mentioned that why would more sodium be outside and potassium be inside? They're both positively charged, the same amount. I mean, they have slightly different molecular weights, and but why would it be different? Well, the brain, the cells actually have an ion pump to force this to happen. They make it happen. The cells want to have the negative potential because that's how cells work. You'll see that they need to have this in order to use the signals that they send to each other. So the way they do that is they force more sodium ions outside, and they force more potassium inside by using energy, ATP, right? Cellular respiration. They use energy via ATPase to move sodium ions outside and potassium inside. And basically this works by moving 3 sodiums outside for every 2 potassiums inside. And that consumes ATP, or which is derived from sugar, right? The power of biological cells. So this is called the ion pump, sodium-potassium ion pumps. Of course, when you eat a banana or something, right, you want sodium, you want potassium and such to be roughly equally distributed all around the brain so that any cell can use it, right? Any cell can you want the gradient to be similar everywhere so all the cells can have the right concentrations and keep the right membrane potential, right? Well, astrocytes, remember astrocytes is a type of glial cell. Glial cells are the support cells, right? Glial cells are support cells for the brain. There's one type is called the astrocyte, which are astrocytes star-shaped as astro, star. And they actually, they have membrane potentials, and they actually take potassium inside the cell, and they, it diffuses within the cell and then moves it to sort of spatially buffer the potassium all around the extracellular medium. So that's one other role that glial cells, support cells, have. So now I'm going to show you parts of a short video that basically shows the membrane potential and the equilibrium potentials for different concentrations of potassium, right? So when you have the cell, you have some potassium inside, and you have a lot of potassium. Should be inside, right? Mostly inside. You have lots of sodium outside, right? And so when you have a situation like this, maybe you have negative 65 millivolts, right? But if you add, right, you just use, add chemicals. You just add extra potassium. You force someone to eat 1,000 bananas, right? And you just add lots and lots and lots and lots of potassium outside. What happens? What do you think will happen? I mean, think about it. We learned about 2 forces, diffusion force and also electrical force, right? So if we have all this high concentration of sodium guys out here, they're probably going to try to go inside the cell, right? And then once they're inside the cell, they're going to try to go back out of the cell. So you're going to find a balance again. and we'll learn that how we'll see how the balance changes when you increase the potassium concentration outside the cell. So I don't know if sound will work here. The video is on pandas, so you can load the video from pandas and watch it along with me if you have trouble with the sound. I'm going to skip through the video so you don't have to watch all of it because some of it is not related to this. The video is about membrane potentials, and they're going to use a crayfish, a type of kind of like small shrimp animal.
And the ionic basis of the resting membrane potential, and to learn methods to measure the membrane potential, as well as to demonstrate properties of synaptic transmission.
So I'm going to explain
in one part
to
of the
you
experiment, the resting
they're
membrane
doing.
potential will be measured while altering extracellular potassium.
So first of all, this here represents the equilibrium potential of potassium and sodium across the membrane is going to be determined by this equation, which is basically related to the concentrations of the ions, potassium ions and the sodium ions. And it's through the log and related to the temperature and other things like that, of course. Some constants.
In another part of the experiment, mechanisms of synaptic differentiation will be investigated by recording stimulant.
I'm going to skip through. So this is the crayfish.
Muscle fiber types.
It's like a sort of shrimp thing.
And
so first what they do is they dissect, or they
remove the legs.
They're trying to isolate some cells, and then keep the cells alive by themselves, just the cells. They don't care about the animal anymore. They just want to get the cells. And so to do that, they're going to cut off the head, basically kill the animal, but they're going to keep the cells alive artificially by giving them sugar and other the nutrients that they need. And the cells will stay alive for a while, even after the animal dies, of course, right? And for animals like crayfish or lampreyfish, you can actually keep the cells alive forever, almost. They're very sort of robust.
Claws of the crayfish to avoid injury.
So first they dissect it,
save the
and they're
abdominal
getting these
portion of the crayfish and dispose of the
muscle
thorax.
cells, basically.
Pointing the scissors slightly using forceps, remove the shell by pulling it with forceps at the top and
They're
pulling it
getting rid
away
of
from the abdomen.
GI,
Cut the bottom of the GI tract at the end of the tail. Rinse the dissection with saline. Abdominal extensor muscles are now exposed.
And so now these are muscles, and muscles are made up of neuron cells, actually, a kind of, they have cells that drive them.
Pour saline solution into the petri dish to cover the apparatus used for intracellular
Of course, it's very small, so they have a microscope and some for dissection. They have a computer to see the membrane potential on, an amplifier to make the currents stronger, because we saw it's like a few millivolts, which is very small. So to see it on the computer, they have to amplify it. They have to make the signal stronger. Some other equipment.
Recordings is shown in this figure. There, into the saline bath. Place the tip of a glass micropipette filled with 3 ml.
Let me see.
Under high-intensity illumination, the M muscle. The electrode should barely be inserted.
So first they're sticking an electrode, basically a wire, usually made out of glass, actually, into the muscle.
Into the muscle fiber. Be careful not to penetrate all the way through the cell. Measure the amplitude of the resting potential. Drag the M cursor onto the trace from the location on the lower left-hand corner of the chart window.
So this is the
Place
membrane
the marker
potential
M on the
as they
baseline
insert
and move the cursor to the lowest
electrode
point on the
inside. So this is
recorded
probably
signal.
the
The
resting
difference
is going to
between
be
the
down
marker
here.
and the active
See how
cursor
it's
is
about
displayed on the upper right side of the screen.
-71.
This value is the voltage of the resting membrane potential. Divide the recorded voltage by the amount of amplification. In this case, 10x amplification was used. Then convert the voltage from volts to millivolts. After recording the resting potential for that muscle fiber, look through the microscope and carefully solutions will be used with potassium concentration.
Okay, so they're going to try using different potassium concentrations for the extracellular fluid. So 40, 60, 80, and 100.
Of 5.4, 20, 40, sodium chloride solution, being sure to cover the preparation completely each time. The effect of raising the extracellular potassium concentration on the membrane potential is shown in this screen capture.
So they added 20. So at the beginning it was the normal one. And so now they're increasing the potassium, increasing the amount of potassium. First, of course, it's outside the cell. But then it slowly will, because of diffusion and electrical force, it will diffuse inside the cell too. So this is the change of the potential as they add higher concentrations. You see it went up. Now they add 40. It's going to go up again. This is the 60. It's going to go up again. 80. It's going to go up even higher. And 100. It's going to go up even higher.
Plot the resting
membrane. So they can
membrane.
plot it. So basically, as they increase, how do I get rid of this? There we go. As they increase the outside, as they increase, that's annoying. As they increase the membrane potential, the potassium concentration, so moving right on the x-axis, this is plotted with a negative opposite, right? So they're going from negative 80 up to 0. So you can see it's the membrane potential is increasing roughly linearly, and this line, the dotted line, is the predicted resting potential based on the equation we saw at the beginning.
Potentials for each concentration. Line up the resting potential at the slope of the hypothetical line.
Okay, so that's all I'm going to watch in this video today. I just wanted to have you guys see what will happen when you increase the potassium concentration. You'll see that the potential, the resting membrane potential changed, right? It went up, which is what you would expect. If we have it here and you have a couple K pluses here and you just add a bunch of K pluses, you make the person eat 1,000 bananas, right? Then because of electrical force and diffusion, more of them are going to go inside the cell. So you're going to have more K pluses inside the cell. If you have more K pluses, well, it's going to make the membrane potential go up, right? Because you have more positive ions inside the cell than you had before. So that's what we'd expect, just based on first principles. Okay, so now we talked about resting membrane potential. You know what is the membrane potential. It's the difference in voltage between the inside and the outside of the cell, and it's caused by diffusion and pumping of ions inside and outside of the cell via channels. Well, actually, the membrane potential changes. It doesn't just always stay at the resting potential. Of course, because the cells wouldn't be doing anything. You wouldn't be thinking at all, right? Your brain would just be stopped, just keeping alive, but doing nothing. Well, it actually turns out that neurons communicate by changing their membrane potential. That's the signal that neurons use to communicate with each other, is changing their membrane potential. And so sometimes in some types of cells, like in your eyes, you have photoreceptor cells, and they change continuously. Like the membrane potential changes a little bit at a time, kind of like this, right? Based on the amount of light that's coming into your eye. Or neuron cells in like small worms or slugs or things might also be like that. However, in a lot of brain cells or other neural system cells, it changes a lot at a time by sending a pulse. It's called an action potential or a spike. It's called a spike because, of course, if you look at the membrane potential intracellular inside the cell, it will look like a spike in the voltage. And often a neuron cell will send many of these. If it will, and the speed of them or their timing or other properties can actually be used to carry information. For example, that volume, the intensity of sound coming into your ear, is communicated along the auditory nerve from the cochlea into your cochlear nucleus in your brainstem. And so you can actually see action potentials, or you can see the membrane potential either inside or outside of the cell. So if I measure outside the cell and I measure the resting outside potential, the extracellular potential, I'll see when the cell fires an action potential. When it does that, you'll see the ions get sucked inside the cell, right? So you'll see all those positive ions. It's going to be sodium ions. You'll see them all go inside the cell. And so you'll see actually outside the cell, because it's losing all the positive ones, it will get more negative. So you'll see like a decrease in the voltage of the outside of the cell. You'll see a negative current because current's being flowing into the cell. So the action potential was first described, or proved by Hodgkin and Huxley, which are two very famous researchers who won the Nobel Prize for Physiology or Medicine in 1963 for characterizing the action potential. And they didn't use human brain cells, they used giant squid axon cells, or the axon of a giant squid, which is very big. It's called the giant axon because it's about 1 millimeter wide, which is a really big, you can see it with your eye. Usually you can't see cells with your eye or axons with your eyes. It's used by the squid to shoot water out, the back, and to move the squid, right? So this is the paper. They described the action potential happening in the axon of this squid, and they won the Nobel Prize for it. The paper was in 1952 in the Journal of Physiology. So basically, action potentials are sort of characterized, prototypical, stereotypical shapes with different phases. So first you have the resting potential of a cell. It's not part of the action potential. And then you have what's called rising phase, when the membrane potential is increasing. It's called the rising phase. Then usually it goes quite a bit above 0 millivolts. So it actually gets positive inside the cell, up to maybe plus 30 or plus 50, depending on the cell and the concentrations. And this is called overshoot. And then it falls. The potential starts to go down again, more negative. And it actually passes by the resting potential before it returns back to it. So that's called the undershoot. So those are the basic phases of an action potential. You have the resting phase, and then rising phase, overshoot, and then falling phase, and then undershoot, and then back to the resting potential, where it can do another action potential. And we can study action potentials or understand them by inducing them, causing them to happen artificially. We can do that by using what's called a stimulating electrode to inject current. Basically, we inject electrons, right, into the inside of the cell. So this is an artificial thing, right? So this is the amount of injected current. We step the current from being, for example, 0. let's say we're using what, like microamperes. And then we go up to using like 30 microamperes or something, right? Is it micro or milli? Wait a second. Negative 65 millivolts. And the equation will be negative V C. And V is IR. So yeah, okay, it should be probably milliamps, not microamps, depending how much you want to move the membrane potential. You don't want to kill the cell by, burning it, frying it. Sorry. Anyways, so you inject a current into the cell, basically force positive, basically, you force electrons to leave. You force electrons to leave the cell, or try to leave the cell by increasing the potential inside the cell. And what that will cause is it will basically cause a bunch of action potentials. And you're also, at the same time, you have a recording electrode to measure the membrane potential, and so you can see the action potentials happening. And so when you inject current, what happens is the cell depolarizes. So this is confusing. This depolarized because, remember, the resting potential of the cell is negative. And being polarized means being further away from zero. So in neuroscience, often you'll talk about depolarizing being an increase in the membrane potential. It gets confusing when it crosses zero because people will still say depolarize, probably by accident. But depolarize usually means increase, more positive membrane potential. And so if we inject a little bit of current, then the membrane potential might increase, depolarize, right, to a different resting value. Because now we've basically changed the electrical force. We haven't injected any new ions, but we changed the relative electrical potentials of the inside and outside. And so ions will move and make a new equilibrium resting potential. And then we stop, we go back to zero, and it goes back to its resting potential. And then we try to inject more current. Now we injected enough to initiate some action potentials. And actually, the more we injecting more current will cause more faster action potentials, up to a certain limit. So let's think about how an action potential happens. How does the flow of ions cause an action potential, the different phases of the action potential? Well, first, your resting potential. We already know about the resting potential, right? During the resting potential, the conductance or the permeability of for potassium and sodium ions, the conductance is much higher for potassium than sodium. And so mostly potassium ions are flowing into and out of the cell. This is because the sodium channels are closed, actually. You can see they're not open to sodium ions moving through. So sodium is the permeability, the conductance of sodium is very low. during the resting membrane potential phase. But actually, during the rising phase, the sodium conductance increases much faster to a higher value than the potassium conductance. And so even though you have a little bit of a little bit of potassium leaving the cell, you have more sodium coming into the cell, causing it to increase, right? If 2 potassiums leave for every 4 sodiums that come in, then you gain voltage inside the cell. And this is because of voltage-gated sodium channels, which I will explain later. First, I'm going to go over the rest of the phases. So the phase 3 is basically the falling phase, right? The voltage-gated sodium channels close pretty quickly. So this is about 1 millisecond, right? Oops, that's hard to see. This is about 1 millisecond or 2 milliseconds. 1/1000th of a second. So after all the sodium ions open and close, then the potassiums are still relatively open. And also additional voltage-gated potassium channels open up. They're slower to open up than the sodium ones. And so potassium flows out of the cell, causing you to try to reach the equilibrium, the resting state. And then later, of course, the pump is trying to, will eventually equalize will bring sodium outside and potassium inside so that we can do another action potential. But first of all, it's trying to normalize the relative voltages, right? And then finally at the end, you're at the same state as you were at the beginning, the resting phase. So those are the 4 basic phases of the action potential. Resting is what we know about. Potassium is moving in and out of the cell to maintain sort of naturally. During the rising phase, sodium channels open, voltage-gated sodium channels open, allowing sodium to quickly come inside the cell, faster than potassium can leave the cell. But then those close and sort of slower potassium channels start to open. Voltage-gated potassium channels start to open, allowing potassium to flow out of the cell and thus reducing the membrane potential back to our resting equilibrium. Well, I mentioned voltage-gated sodium channels, and basically this is a special type of channel that is normally closed. Normally sodium cannot flow through. But when the voltage of the inside of the cell increases enough, the shape actually changes to allow, sort of like opening the door, to let sodium flow through it. And this happens very quickly, within less than a millisecond. This is called, since it's gated, the door is closed when the voltage is low, and it's open when it's high. It's called voltage gated. In other words, it's gated. The door is based on the voltage. And so when the depolarization happens, the membrane potential goes up, then the conformation of these proteins changes and allows, so see it's closed here. The ions can't flow through. But when it moves, then the conformation changes, allowing sodium ions to flow through. That happens at about negative 40 millivolts inside the cell. And so here's an overview of basically you can see the current change when individual sodium channels are opening and closing. Of course, the real situation for action potentials is a little bit more complex. I talked only about up and down, but actually, of course, there's this undershoot we talked about, right? And this is because I mentioned that the potassium are a little bit slower to open. than the sodiums. The sodiums open and close very quickly. The potassiums open less quickly, and then they stay open longer. And so first let's talk about the sodiums. They open very quickly, so it looks like this, right? This is a positive inward current, right? Current to the inside of the cell. This is what increases the membrane potential. You can see it happens very quickly in this short time period. But the potassium channel, which open, they start opening later. And you see they stay open longer. These are causing the outward currents. But then if you add these together, you sort of overlay the sodium and potassium, you can see why you get this overshoot, this undershoot here that lasts longer, is because basically the sodium channels open, potassium channels open, and that balances things out, and that should go back, here. But actually they stay open longer, and so you have a net outward current, a net negative current going outside that lasts longer than the action potential, and that's what causes this undershoot. Because sodium permeability conductance is low, and the membrane potential, it tends towards the potassium potential, equilibrium potential, because potassium is the only thing that can flow into and out of the cell. So it has a more influence on the membrane potential of the cell. Okay, so that's all. That's how action potentials work. That's why they have the shape that they have. So now, just quickly, sort of for fun, I want to ask if you know any poisons, any nervous system poisons, neurotoxins. Can you think of any famous ones? Well, we're in Japan. Fugu. Actually, fugu are poisonous, and they're poisonous because of a neurotoxin. They actually have a toxin called tetrodotoxin, TTX. And TTX is very poisonous because it blocks sodium channels from the outside. It stops sodium from moving, which stops neurons from having action potentials. So basically your brain stays in its resting potential all the time. It stops talking to each other. And that causes you to die. Okay. Finally, just 3 or 4 slides. I'm going to prepare for the next class by talking about how action potentials, AP, travels along the axon to communicate to the next cells. Today, we learned about membrane potential, which is the voltage difference inside and outside the cell, right? Well, we thought mostly about the cell body, the soma of the cell. But actually, everywhere inside the cell has a membrane potential. There's a difference from the inside and the outside everywhere. In the dendrites, right? In the dendrites, in the soma, the cell body, and there's also in the axon. It has a resting potential, and it can have action potentials. And axons are what send signals to other brain areas. And we want that to happen fast. Well, action potentials actually start often at a special part of the axon, or of the close to the cell body, called the axon hillock, the root of the axon. And this is where there's actually the most voltage-gated sodium channels. So it allows the action potential to be initiated because it rises faster than the other parts of the cell. And then, if you think about it, if this is the cell here, right? This is the hillock. Then, when you change the membrane potential here, it goes up, right? It takes a while for the voltage to flow along the axon. But as it flows along the axon, the same thing happens, right? You have an action potential here, just this little part of the axon. Then the current, the voltage will increase, which will cause a current to cause the next part to increase its membrane potential. And that will activate the voltage-gated sodium channels, which will cause an action potential here, right? Which will happen a little bit later, right? It'll take about a millisecond for that to happen. And then this part has higher voltage, and it will flow along the axon, and that will cause the next sort of section of the axon to have an action potential. And that will keep happening. So the action potential will flow along or travel along the axon until it gets to the end. And they only go one direction usually, because after it passes this area, right, you'll be in this time period where there's no sodium outside the cell. There's too much potassium inside the cell. So that cell's kind of tired. It's called a refractory period. For a couple different reasons. The concentrations, and also there's just because of the kinetics of the channels also cause this refractory period. So that's why the signal only usually flows in one direction. Of course, if you somehow injected current right here, it would flow both directions if these weren't in the refractory period. Of course, this is the situation for an unmyelinated axon with no fat surrounding it. But it's very inefficient because you have to basically create an action potential at every little section of the axon, right? Which takes a lot of energy. And also it's very inefficient because the current is flowing into and out of the cell and it's diffusing all over the place. It's very inefficient. So a lot of a lot of vertebrate axons are wrapped in this fat, this myelin, to insulate it. And that lets it act more like a wire, basically. It lets the current flow inside without having to have action potential to keep it alive. So this prevents leakage of ions and lets the current move along the axon. It also allows for what's called saltatory conduction, where the action potentials are only recharged at these nodes where there's no myelin, basically between the different oligodendrocytes. These are called nodes of Ranvier. And so it's the same thing. Basically, it moves along. First it happens here, the current moves. Current, is moving here, and that causes this to be higher potential, and that causes an action potential here. And so that's the conduction is the same concept, but the distance between when it needs to sort of recharge itself is increased because of the increased resistance caused by the insulation caused by the myelin sheath. And that helps a lot. That makes the speed increase a lot. And you can see the difference. You can actually measure how fast an action potential flows through an axon. And so in humans or vertebrates, you have, for example, in humans, you have proprioceptors or skeletal muscles connected by axons through the spinal cord up to the brain. These are called alpha or A alpha that travel from the skin, or if they're from the muscles, they called group 1 axons. And they're very thick, 13 to 20 micrometers. Remember, the squid was 1 millimeter, so it's 1,000 micrometers. So they're 100 times smaller than the giant squid axon, which was unmyelinated, not myelinated. But these guys' speed is about 80 to 120 meters per second. So if I'm, 2 meters tall, very tall person, 2 meters tall, right? That will take one, let's say 2 divided by 120 would be 1/60th of a second, or about 20 milliseconds to travel from my head, or from my skin to my feet to my head, right? There's also other types. So a beta type here is mechanoreceptors, have a little bit smaller. They're also myelinated, and they have a speed of 35 to 75. There's pain receptors, which are these delta receptors that sense pain or temperature, and they're smaller, and they have a slower conduction speed. And finally, you have these things called C fibers, which are unmyelinated, and they sense pain or temperature or itch. And so you can see there's 2 types of fibers that actually sense pain. And that's why if you ever hit your foot, you hurt yourself, you actually feel like 2 waves of pain. First you feel like, pain, oh, it hurts. And then later you feel pain again, right? And that's because actually first is caused by these guys coming from your feet, to your, or whatever. First, the delta ones go faster, and that's this part. And then the C fibers go slower, and so that's what causes the second wave of pain you feel. Okay, so finally, just one thing is I'm going to talk about another sort of recent method for inducing action potentials. So historically, the only way that we could make action potentials was to electrically stimulate these neurons by injecting current with a wire, with an electrode. But in the last less than 20 years, based Carl Diesseroth's work, they actually they modify the DNA of the brain cells to include DNA from algae or microbes that are sensitive to light, called rhodopsins, or opsins, basically, to include opsins, which are molecules sensitive to light. And then you can stimulate the brain. You can cause action potentials or cause decrease in midbrain potential by shining light on the brain. So here's pictured a laser light going into this rat's brain. And so there's different types of opsins that you can use. Channel rhodopsin 2 is an example that's sensitive to blue light, and it opens sodium channels. So basically it forces action potentials by depolarizing the cell. Another type is halorhodopsin, which is pulled from the genes of single-cell microbes, and then responds to yellow light by opening and letting the chloride ions pass into the cell, which hyperpolarizes. So it makes the cell more negative, and that inhibits the neuron. Okay, so that is all for today. I'm going to review really quickly. First is for electrical signaling to happen in the cells. You need to have 2 concepts. The resting membrane potential. So you need to know what is a membrane potential. And then you also need to know about what is the action potential, which is the spike when cells are activated over a certain threshold. And for these to work, these are both caused by sodium and potassium ions and the channels that allow these to pass into and out of the cell, via either passive leakage channels or by special voltage-gated channels or by ion pumps, right? And so that's how a signal, the signal is the change in membrane potential, is sent along the axon, propagated along the axon. And then when it gets to the end of the axon, then actually there's a chemical signal that is sent to the next neuron. And that's what the lecture next week will focus on.